Welcome students to this section. Now in this section I'll be talking to you about DNA replication. So let us see what is this. Now there is a central dogma of molecular biology. What does it tell? DNA upon converting back into DNA is replication, DNA to RNA is transcription, RNA to proteins is translation. This is the central dogma of whole of molecular biology, that DNA can replicate itself, can form a new DNA. Why do we need to replicate? What is the need for replication? We know that cells divide, cells have growth, they have repair, they have maintenance. There is need to replace cells sometimes. So because of all these reasons, a cell can keep on dividing. Now, before a cell is going to divide, it needs to double all its cell structure, whether it is the cytosome, whether it is the subcellular organelles, everything it needs to double it, and including the DNA. So, and especially their genetic information. They have to double the cell structures, organelles, and their genetic information. So, this process of exact copying, duplicating the genetic information from the parent DNA is known as replication, where a DNA strand is made into an exact duplicate of it is known as a Xerox copy of it is known as replication. Why don't we call it as a Xerox copy? The reason is there is slight difference between all those things. How the copy is going to take place. If you look at the model about that, we'll come to know why we call it as replication. So, why is it essential? What is the purpose of replication is to see that the provision of progeny with the genetic information possessed by the parent. Whatever the parent cell or the parent is having, whatever genetic information, that has to go and reach the progeny. It has to be having the same genetic information as that was there in the parent. So during each cell division, each daughter cell, both the daughter cell should get an exact copy of the genetic information. Whatever gene is present in the mother cell should be present in both the daughter cells. Both should contain the same genetic information of the mother cell. So, replication of DNA is a means by which genetic stability is kept on, genetic stability is maintained between organisms and species. That is why even if the cell is keeping on growing and growing, cells are dividing, repairing, everything is happening, the genetic information will remain the same in a new cell also. So, because of this, it is possible because of DNA replication. When does replication occur? So for this you will have to have a bit of background about the cell cycle. You know that the cell cycle is made up of different phases, like the G0 cell cycle, the G1, then the gap, that is gap 1, then S phase, then M phase where mitosis is taking place. There is a gap 2 also. In some cells they go into the quiescent state, they don't come back to the G1 state or to the synthesizing state. So this information about the cell cycle is essential so that you will come to know where this is taking place. So when replication should take place? It should ideally take place before mitosis, because at the time of mitosis that much genetic information, the doubled genetic information should already be available to the cell. Only then it will go into mitosis. So when does this take place? It is known as the S phase of the cell cycle or the synthetic phase of the cell cycle. So S for synthesis phase of the cell cycle is when the chromosomes, all the chromosomes are getting replicated. For this requires DNA synthesis, and also at the same time it requires histone synthesis. Why? Histones are necessary to pack the chromosomes, pack the DNA into chromosomes. We need histones. So they will pack whatever newly synthesized DNA is there, to pack them, make it compact, we need the histones. So during the S phase, not only is DNA replication taking place, at the same time histones are also going to be synthesized, so that by the time the cell is ready for mitosis, both it has the DNA, it has other subcellular organelles, and also the histones, all in the form of chromosomes. Now it is ready to divide. So for this sake, both DNA replication and histone formation all go hand in hand. Now, what are the general features of replication? First and foremost, replication is semi-conservative. What do I mean by semi-conservative? What else could it have been? One possibility could have been that if the mother cell DNA, the same DNA was present in the daughter cell, then you could have called it as conservative. Otherwise, the one more thing could have been, the whole thing could have, or both the strands could have made a new DNA. Both of them could have been the new. So if I'm going to show the new in the form of a different color, so if both the daughter strands were having both newly synthesized DNA, it could have been a different type of model that could have been happened. But this also doesn't happen in our body. In our body, what happens? In our body, we have what is called as a semi-conservative DNA replication. What do I mean by semi-conservative? By semi-conservative, it means each old or the original strand will go to one daughter cell. So the one daughter cell will have one original strand, the other daughter cell will have another original strand, and the new plus the daughter needs both the strands, right, for double stranded, and one newly synthesized strand. So this means this is a semi-conservative model. Why semi-conservative? There has been conservation of the original DNA, but complete conservation? No, only partial conservation. Why? There is one new strand and one old strand. So this is the new strand and this is the old strand. So each daughter cell will contain one old and one new. This is known as the semi-conservative model. Now, how did we come to know that this is what is happening? One new and one old. How did we come to know of that? There is one classic experiment done by Michelson and Stahl, and this they used what was called as labeled substances, like nitrogen labeled substances N14, N15, and then they found out that when this cell culture was looked for the DNA, the daughter cells had a mixture of both N14 and N15, and that is how they came to know that within the body DNA replication, or not only within the body, whether it is prokaryotes or eukaryotes, the DNA replication is semi-conservative. DNA replication is semi-conservative. So that is a main feature of replication. This is the semi-conservative model. The double-stranded DNA contains one parental and one daughter strand or the new strand following replication. This was proved by the Michelson-Stahl experiment. What else is required for replication? What are the requirements? So, this is DNA synthesis. So, what are the substrates required? Deoxynucleoside triphosphates, or what we call as deoxy ATP, deoxy GTP, deoxy CTP, deoxy TTP. All the triphosphates have to be there. The purine and the pyrimidine AGCT in the deoxy form are required before the start of replication. What else is required? The mother parent DNA is required. Each strand is going to act as a template for the replication of the new strand. So, template is required. Why do we need the template? So that, remember, during this process, what is happening? The genetic information that is present in the parent has gone to the daughter without any mistakes. So, for this, you need a original strand is going to act as a template, and base pairing has to occur. Only then genetic information will go from one strand to one cell to the daughter cell. So, the template is required for the addition of appropriate complementary nucleotide to the newly synthesized DNA strand. Enzymes are required, and more importantly, to differentiate it from transcription or translation, DNA replication requires a primer. DNA replication requires a primer which has been made from RNA, and there is a specific enzyme which catalyzes the synthesis of this primer, and this is known as the primase. Primase is a DNA dependent RNA polymerase. Polymerase is something which is going to polymerize. Here what is being polymerized? The deoxyribonucleotides are being polymerized. But the primase is something which is going to use DNA as a template but synthesize RNA. Instead of synthesizing DNA itself, so if this is the DNA, just for the sake of convenience I have used, it will synthesize RNA. This formation of primer is essential in replication. I'll talk more and more about all this as we go ahead. Let us look at which are the enzymes that are essential. And before that, we'll start with the prokaryotic and the eukaryotic enzymes. Now, what is the difference between replication in prokaryotes and eukaryotes? Please remember, in cells like bacteria, the DNA is circular, whereas in eukaryotic, the DNA is linear. So there is going to be some amount of differences between prokaryotic and eukaryotic replication. However, please also remember that the eukaryotic DNA is a huge DNA, whereas prokaryotic is a very small DNA. Taking all this into consideration, there are going to be differences between prokaryotic replication and eukaryotic replication. So if you look at the prokaryotes, the RNA primer will be around 50 nucleotides, quite long. Whereas eukaryotes, it is approximately 9 nucleotides. Now the DNA polymerase, the enzyme that is required for the synthesis, polymerization of deoxyribonucleotides, is known as DNA polymerase. Now in the prokaryotes, there are 3 types of this DNA polymerases. I'll talk more about each of them. They are known as Pol1, Pol2 and Pol3. Whereas in eukaryotes, there are mainly 5 types. They are called as alpha, beta, gamma, delta, epsilon. So DNA polymerase alpha, Pol beta, Pol gamma, Pol delta, like that the names go on. Now if you look at the origin, the site where replication has to take place, prokaryotes have a single origin. That is, only at a single base within the DNA is going to act as an origin. Whereas eukaryotic, imagine such a huge eukaryotic DNA, so many chromosomes, is it enough to have one origin? No, not enough. So that is why in eukaryotic DNA replication, multiple origins are there. That is the main difference. Now there is what is called as a length of the Okazaki fragment. I'll tell you what it is. Right now, remember that in prokaryotic it may be 1000 to 2000 nucleotides, whereas in eukaryotes it is approximately 200 nucleotides. And the rate of replication is more in prokaryotes. Around 500 nucleotides are added per second, whereas eukaryotes 50 nucleotides are added per second. Why? In eukaryotes it is very essential that proofreading has to take place. So the polymerase enzyme is having both proofreading and adding. So every base that it is going to add, it sees to that it is the right base that it has added. So it keeps on proofreading and adding, and so it is a slow process. They do not want a single mistake to happen when we are undergoing replication. The eukaryotic replication, there is no scope for mistake. Mistake should not happen. Why? Even a single base deletion or a single base mismatch can lead to a deleterious effect, can lead to different consequences and different diseases. So it is very essential. So eukaryotic replication takes place slowly, 50 nucleotides per second. Prokaryotes are not so bothered. They have mass production like that. They have a rate of replication, huge rate of replication. Now let us go to the different enzymes and proteins involved, and then I will tell to you how replication is taking place. Now as I said, lot of enzymes are required, lot of proteins are required. First I will give you a list of these enzymes and then I will tell you the exact procedure. Now DNA polymerase is an enzyme, and what is its function? polymerization of deoxynucleoside triphosphate, DNTPs. Whatever deoxyribonucleotide is coming in, it is going to polymerize them. Is going to see that a phosphodiester bond is formed between one polymer, one nucleotide and another nucleotide. So this polymerization is done by the enzyme DNA polymerase. Primase, as I said, is the one which is a DNA dependent RNA polymerase. So what is the difference between DNA polymerase and primase? DNA polymerase is going to polymerize deoxyribonucleotides, whereas RNA polymerase or the primase is going to polymerize ribonucleotides. The difference is in the sugar. So primase is responsible for the formation of RNA primer. There are what are called as helicases, which are responsible for unwinding of the DNA. Remember, the DNA is forming a double helix. So somewhere or the other, each of the strand is going to be moved out. So when you are going to move out, you need to have something which can remove this helix, and that is known as a helicase. Helicases are responsible for unwinding of DNA. Whenever you have got a spiral thing, if you try to remove it, what will happen? There will be formation of supercoils ahead. This side or that side, there will be supercoils when you try to remove something which is highly coiled. So to remove the supercoils, we need certain more enzymes, and these are called as topoisomerases. They relieve the torsional strain or relieve the supercoils that may be formed. Then we have DNA ligases. They are responsible for ligating the nick that is formed. Suppose there is wherever there is a DNA cut, there is a difference in the two DNAs are together. To ligate them, we need the DNA ligase, joining of DNA fragments. And this is very important during the Okazaki fragments. Then we have what is called as DNA A protein. DNA A protein is to recognize the site of origin. Now where to start? So much DNA is there, so big DNA is there. Where to start? The DNA A protein will know how to search for the site of origin and go and bind there. So DNA A protein is to bind at the origin and cause a local unwinding at that origin. So DNA A protein. Single stranded binding proteins. Minute the DNA from double stranded becomes single stranded, there are enzymes which are ready to pounce on it and eat up that DNA. So what will happen? The DNA will just get digested. So to prevent this, we need certain proteins that are necessary to bind to the single stranded DNA. So minute the double stranded DNA is unwinding, it has formed a single strand, it is susceptible to attack. To prevent this attack, we have certain proteins which will come and bind to this single stranded DNA, and these are called as SSPP, single stranded binding proteins. They will prevent the premature forming of double stranded DNA and also they prevent the attack of nucleases on the single stranded DNA. So among all this, these four are these till DNA ligase are enzymes. The remaining two are proteins, DNA A protein and single stranded binding protein. Now let us look at what are the features of DNA polymerase. I said DNA polymerase is required. So let us look at that different DNA polymerases. So if you look at the prokaryotic DNA polymerase, they have three types: Pol 1, Pol 2, Pol 3, out of which DNA polymerase 3 is responsible for replicating. The most important enzyme for the whole replication process is DNA polymerase 3. What does DNA polymerase 1 do? It removes the primer and fills the gap. It has what is called as exonuclease activity. I'll be explaining it more to you as we go ahead. So DNA, just remember for this sake that Pol 1 removes the RNA primer. Wherever there is a RNA primer, it can eat that primer, removes that primer, and it fills the gap. DNA polymerase 1. And DNA polymerase 2 is responsible for DNA repair. What we call as proofreading and DNA repair is done by DNA polymerase 2, and it has 3', 5' exonuclease activity. I'll tell you what these also mean. Now, what is the eukaryotic DNA polymerases? What are its function? As I said, there are so many of them. Now, the alpha eukaryotic DNA polymerase alpha is responsible for gap filling and synthesis between Okazaki fragments. Beta is responsible for DNA repair. Gamma is responsible for mitochondrial DNA synthesis. Delta is responsible for the leading and lagging strand synthesis. This is the most important enzyme. Epsilon is responsible for DNA proofreading and repair. Now, there are other DNA polymerases also in the eukaryotes, and these are also called as bypass polymerases because they bypass the area of DNA damage and continue with replication. However, as of now, from an MCQ point of view, from a knowledge point of view, knowing that these are important is enough. Alpha, beta, gamma, delta and epsilon. These are the different eukaryotic DNA polymerases. Now, there are different steps in DNA replication: initiation, elongation and termination. What are the things that are happening during this? I'll talk about it in my next session. Thank you.